I am ecstatic that the samples were transferred and that some resources were provided for their care. The biological monitoring program is impressive in its temporal and spatial scope. So in that sense, because they've been doing this for so very long and they've been using, let's say, a reasonably constant sampling design, and there's so many of these fish larvae that the potential is truly staggering. Having worked with the Normandeau samples, I realized that there was an additional opportunity that could do what I viewed as a more decisive job of looking at the impact of zebra mussels by looking at the feeding success of larvae before and after the arrival of the zebra mussels by two means. One, looking at diet, stomach contents, and the other looking at the condition of the larvae, the mass per unit length, essentially, as an indication over a longer time scale than the stomach contents, how successful they were at finding food. And our predictions were that the zebra mussels, of course, were filtering the water, thereby depleting the phytoplankton, thereby depleting the zooplankton, and thereby impairing the feeding success of larvae. We knew that some species in the Hudson River had suffered many years of reduced recruitment and that year class success was decreasing. In particular, we thought that the American shad was going to be one good target species for this analysis. And we threw in also striped bass larvae as being perhaps another good opportunity to look at the potential impairment of feeding success by zebra mussels of another target species in the Connecticut River. So that's the reason why, one of the reasons, I guess that probably explains most of the samples we have now in my lab from the monitoring program, was our effort to document over the period of the zebra mussel occupants of the estuary what kind of changes there have been in the fish larvae. For the longest time, those samples were very difficult to get. They were not being watched over for understandable reasons, I would say. And the data were available, but you had to, let's just say, jump through some hoops. This is going to be true of any monitoring program, and there's a huge spectrum of attitudes about monitoring. And one of them is that, and I'd say this is probably the case for most monitoring programs, that there's, many of them are designed without necessarily thinking about the connection between what the monitoring program is for and what the objectives are. I will say that it has been definitely working with the Hudson River Foundation and working with the utilities in terms of getting certain data sets, getting the samples, and in working with the state, has definitely sensitized me to the, I'd say, colossal complexity of doing a lot of I would imagine, in any kind of grand scale effort that involves helping science inform policy. Because I like to think that that's what the Hudson River Monitoring Program has done, is science in the service of wise environmental policy.